Hi, welcome to NPTEL Animal Physiology Extra Live Session Week 3. So, this week basically we have learned more about one of the system as we are going in the order starting from the um, we saw the hierarchical session starting with the cell, tissue, then organ system. So we are going through different types of organ system one by one. So first we were dealing with muscle. So there are different types of muscle. Muscles can be classified according to the depending on the force generation, as well as there are different types of classification. So first in the first lectures are explained about based on the force generation, muscle can be divided into three types: skeletal muscle, cardiac muscle, smooth muscle. So more amount of force is generated on skeletal muscle. Skeletal muscle we are able to move like this. Whatever the movement is happening through that is through skeletal muscle. So skeletal muscle is most force is developed in skeletal muscle. And there is smooth muscle and cardiac muscle also. The cardiac muscle is, as the name indicate, cardiac, the word related to heart. So in the heart, it's always working. Lub, dub, lub, dub. So there is limited activity, but it's always working. Hi, Maithili. So it's making lub, dub sounds. So that's also indicate that yes, the activity is there and smooth, the function of cardiac muscle is there, but it is lesser compared to the skeletal muscle. In smooth muscle also, it's like the blood vessel, the vessels that is seen in our intestine, all these are smooth vessel. So smooth muscle, we know the peristalsis movement. So very less force is required there. So that is about smooth muscle. So let me share the screen. Yeah. So as I said, the class started with introduction of muscle, then about skeletal muscle formation, followed by anatomy of the skeletal muscle, and then followed by contraction in skeletal muscle, finally function of actin and myosin. So coming to the introduction of muscle, as we already discussed, in muscle it can be classified differently, and there is axial and appendicular muscle. So based on the force generation, now just we discussed, so I'm not discussing anymore. It can be divided into three types. So here you can see the figures of skeletal muscle which can develop more force, and then cardiac muscle, then smooth muscle. Depending on the classification, as like muscles can be divided into force generation subtypes. It can be also divided into microscopic detail, structural due to the structural variation also. So here you can see skeletal muscles looks like there is some striation present. In cardiac muscle also we can see, but in smooth muscle we cannot see that. So they are non-striated muscle, as well as skeletal and cardiac muscle are striated muscle type. So let's go to the some questions related to this part. Then we'll discuss some question next to the second part, then followed by some more question. Like that, we will be going today's class. Yeah. So first question is, which of the following muscle typically operate under voluntary control? Option A, smooth muscle. Option B, cardiac muscle. And option C, skeletal muscle. So we just now discussed it's about voluntary control, right? If I want to move my hands, it's voluntarily I want to take something. So it's voluntarily I'm doing that. So in all the voluntary type of control, it's related to the skeletal muscle. Okay. So the correct answer is option C. Second question is, which of the following muscle have the ability to cause blood vessels to dilate or constrict depending on external condition? So question is, cause blood vessels to dilate or constrict. So I said just now, so the blood vessel movement is involuntary. and it's about blood vessels to dilate or constrict depending on the external condition. So the muscles that is present in the blood vessels and movement related is option A, smooth muscle. So correct answer is option A. Okay. Now, which of the following muscles do not consist of striation? So just now we saw the figure and we already discussed depending on the microscopic anatomy, structural details, we can again divide the muscle. So non-striated muscle, as we just now discussed, it is smooth muscle. They don't have any striation, so they are non-striated muscle. So option C is the correct answer. Now, second lecture was about mainly the skeletal muscle formation. As we all know that each organ system, everything consists of the hierarchical organization starting with the cell, then tissue. Even before cell, there is molecular level of organization, then cell, then tissue, then organs, then organ system. So like that, if we are going to the structural hierarchical organization of muscles, then we can see there are individual cell types present called as myocytes. So anything related with myo is related to muscle. So myocytes, site means cells. So myocytes are the basic and functional units of cells that are present in muscle. These different types of myocytes or the first muscle precursor cell or satellite cell that will different types of cells come and join together to form myotubes. So then myotubes again combine together too. So myotube we can see there are different multiple nuclei and an elongated cell type. Okay, such different types of tubes join together to form myofibers. So each myofibers again combine together to form a large functional unit called muscle fibers. Okay. Now the question is, what is the role of guidance cue in the arrangement of muscles? So what is the role? Because uh, you think like this. How these cells are able to come. Okay, now cells understood. I have to go to that location. There I have to divide and I have to form tissue and I have to form this like and again I have to go and in this manner. So all these regulation to how to where to go to where to build up to how to form everything is regulated through the help of some guidance cue. So guidance cue helps in the muscles to come and arrange at the particular pattern. Okay, that was about lecture 2 introduction. Then lecture 3 anatomy of skeletal muscle. So in the anatomy, hi. So at 6 I started the class since so I have not waited for no one joined that time. That's why I started class. So I was just recapping through all the lectures. What is the um, general things as taught. So I just covered now lecture 1 and lecture 2. Lecture 3 was about anatomy of skeletal muscle. So in the anatomy of skeletal muscle, we again go into deep into each muscle fibers and show the structure. Okay, so based on the microscopic observation, as discussed in the first sometime before also, muscles can be classified into striated muscles as well as non-striated muscles. So I showed that picture also like this one. So striations are present in skeletal muscles and cardiac muscle, but there is no striation present in smooth muscle. So we saw that and we also saw what are the microscopic structural details of a muscle. So if we are taking like this, then bone and then bone and muscle are connected by tendon. So if you are looking at each individual muscle fiber, each individual muscle fiber can be called as myofibril. So this is myofibril Each myofibril is again surrounded by, so this is important to know where is endomysium, perimysium and epimysium. The whole muscular muscle is covered by epimysium. A group of muscle fiber is covered by perimysium and individual myofibrils are surrounded by endomysium. So remember, individual muscle fibers are covered by endomysium, group of muscle fibers are surrounded by perimysium, and a muscle total this structural is covered by epimysium. So please keep this in mind, and there is multiple questions coming at this stage. So please don't get confused. Okay. So let us discuss some more questions from this part. My screen is stuck. Yeah. So that's what we were discussing, and I hope you remember last time we discussed about osteoblast, osteoclast. Osteoblast is always for the formation of bone, and osteoclast is the removal of or making a cavity. Osteoclast is required. Like that, you remember myoblast is always required for the formation of muscles. Okay, so what happens? Myoblast fuse together to form myotubes like this, which will differentiate to become a complete muscle fiber. Okay. So if you are looking at the order of how muscles are formed, it will start with the stem cell or muscle progenitor cell will give rise to myoblast. Myoblast will give rise to individual myocyte. They will come and migrate and settle down, and together they will form muscles. Okay. So let's solve some questions. Dash is the connective tissue that surrounds the entire muscle. So this part is very important. Please keep in mind that um, we have to go through this session very clearly. Please don't get confused about endomysium, epimysium and perimysium. So we will go one by one, and after dealing with this question, you will be more clear regarding what is endomysium and what is epimysium and what is perimysium. So question is, dash is the connective tissue that surrounds the entire muscle. Just now we discussed. Please type the answer in chat box, then we'll discuss the answer. Okay, I will show the question again. Dash is the connective tissue that surrounds the entire muscle. Shreya and Veeramuthu, please type the answers, or you can open the mic and say the answer also. I will wait till 6:19, then we'll start discussing. Okay. I will be happy if you guys are joining with me and communicating and telling and typing the answer. Otherwise, also it's fine. I can understand. But if you discuss, sorry. Hi. Hi, Shreya. Hi. You can hear me, right? Hello, you can hear me, right? Okay. So, do you know the answer? We are discussing the first question. Dash is the connective tissue that surrounds the entire muscle. It's fine. Uh, okay, the answer is already there. Let me go back. Yeah. Dash is the connective tissue that surrounds the entire muscle. Just now you saw the answer, right? Now at least you can say the answer. So you can remember by seeing this figure, right? So it is the question was entire muscle. So entire muscle, the most outer layer is epimysium. Okay, so the correct answer is epimysium. Now second question is dash is the connective tissue that surrounds the group of muscle fibers. It's just group of muscle fibers, not the entire muscle. So the layer that comes below that. Now we know entire muscle is covered by epimysium. What is the connective tissue that surrounds the group of muscle fibers? Group of muscle fibers. Any clue? So the correct answer is, should I go back to the figure again? This one. Group of muscle fiber is surrounded by, what is this written on the screen? So group of muscle fiber is surrounded by perimysium. Okay. Yeah. So entire muscle is surrounded by epimysium. Group of muscle is surrounded by perimysium. And if we are third question is, if we are looking at the individual muscle fibers, what is the connective tissue that surrounds individual muscle fibers? Is it endomysium, epimysium, or perimysium? What will be the correct answer? Since we already know what, yeah, correct answer is endomysium. Yeah, good, good. So, entire muscle is covered by peri, and group of muscle fiber is connected by covered by epi, and a connective tissue that surround individual muscle fiber is endo. Okay, this is endo, peri, and epi. Sorry, I said oppositely. Entire muscle is surrounded by epimysium. A group of muscle fiber is surrounded by perimysium, and individual muscle fiber is surrounded by endo. Endo, peri, epi. Yeah. Now, fourth question is, a tendon is an extension of which one of the following? Option A, endomysium and epimysium. Option B, endomysium and perimysium. Option C, endomysium, epimysium, and perimysium. What is the correct answer? So, what is tendon? Tendon is in this figure. Maybe my laptop is stuck. Yes. You showed this, right? Tendon is the one that connects bone to the muscle. So, if the question is, what is the correct question? Tendon is an extension of which one of the following? Uh, so, all the tendon is an extension of total muscle, right? So, which is covered by endomysium, epimysium, and perimysium. So, correct answer is option C. Okay. Yeah. Let's move to the next page. And finally, fourth lecture was regarding contraction in skeletal muscle. So we know the structural hierarchy. Everything starts with the cell. Even inside the cell, there is lot of molecular players which regulate all the function. If I am able to move my hands like this or do like this or anything that voluntary movement I am able to do with my skeletal muscle, it's because there is some molecular players that is present inside each cell types. So, for example, if you are looking in the skeletal muscle, then if you are going and dividing and seeing if the individual cells, myocyte, and looking inside again, we can see some proteins. Okay, so important protein present for the muscle sliding is actin as well as myosin. Okay, so this actin and myosin and how they are arranged is the vital players that play a key role which helps to enable the force production in skeletal muscle. Okay, so basically whatever the muscle is doing to do the movement and all is basically due to the molecular proteins present in them called as actin and myosin. And also not only the muscular activity, everything has electrical activity also. And in this class are also explained there is we can divide the muscle types the types based on electrical activity also. So whichever the types has electrical activity cell types, that is known as excitable cell, and whichever doesn't have the electrical activity, that are non-excitable cell. For example, our muscles are electrically excitable and our neurons are electrically excitable, but there are other cell types which doesn't have any electrical activity. Okay, so muscle and neurons are electrically active cells. So why it is important? Because we know my brain is telling, suppose I put my hand accidentally in some fire. So my brain is giving the instruction, Harsha, this is dangerous, withdraw the hand. So that I am getting, right? So my neuron cells are giving the signal to the muscle cell. So that's why they are electrically excitable cells. Okay. And here also we have given a figure. So these are where we discussing sarcolemma is the membrane covering the muscle. Then there are nucleus, mitochondria is required as the movement is there. In order to produce movement, there should be source of energy. And we know powerhouse of energy is mitochondria. So in muscles, we can see a lot of mitochondria because they have to produce ATP. And myofibril is the individual fibril units. Okay. Now, if we are again looking at this pattern, we can see there is some Z stripe structure. So, this Z stripe structure is known as Z line. This Z line is Z line constituting zone is the I band. Then we can see the thin filament called as actin and thick filament known as myosin. Okay, basically this Z line is constituted by the actin filament. Thick filament forms a central line like this, which is known as M line, and the zone between the thick band is known as H zone. Okay, here you can see between actins in the relaxed state, we can see the just constituting the myosin. There is a central zone that is known as H zone. And from this total, the distance between two Z line is known as a sarcomere, which is the functional unit in sliding contraction theory. Okay, so please keep this in mind. Thick filament is myosin, thin filament is actin. So what is sliding filament theory? So the thick filament of myosin moves in between the thin filaments of actin, that is sliding filament, or slides through the rails of the actin filament, that is sliding filament theory. Okay, please keep all these things in mind. Now we will go to next slide. My screen is stuck sometimes. Yeah, this where we discussing what are the steps of sliding filament theory. So what is sliding filament? They where they occur? They occurs in sarcomere region as we just now saw. So what is sliding filament? The myosin filaments ratchet over actin filament contracting the sarcomere. And as we discussed, there are two bands, I and H bands, which compress and expand to facilitate the movement. And the myofilaments do not expand and contract on their own. Okay. So what are the steps? First, there is some calcium coming. So always remember that calcium is released from sarcoplasmic reticulum. It is same as endoplasmic reticulum, which is in the muscle. That's why it is known as sarcoplasmic reticulum. So they release some calcium ions. So when the calcium ions released, after calcium ion, there is some binding of calcium binding units in actin that we discuss in the next chapter. There is some three different units of actin and myosin. So when myosin heads hydrolyze ATP, So they become reoriented and energized. As a result, what will happen? Then only myosin is able to bind to the actin, which forms the cross bridges like this. So which require the formation of after ATP binds, then ADP phosphate, then myosin heads binds like this, which form the cross bridge. After the cross bridge formation, they will pull toward the center of the sarcomere. That is known as power stroke. So after that, myosin heads binds ATP and cross bridge detached from the ATP. So this is the four steps in sliding filament theory. We can see a video also so that we can remember this. Again, yeah. Yeah, that about that video. So there is some extra information told how that calcium is released because of acetylcholine released from neuronal fibers. This we will be seeing detailedly in the coming classes. Sir will explain in very detail, so we can go through that time in the next lecture onward. But I hope now you will understand how the basically sliding filament theory works. Okay. Now I have also given, since the molecular players are actin and myosin, it is important to know what are the differences between actin and myosin. So we understood they are basically proteins, but by definition, actin is a group of globular protein and myosin is a subfamily of motor protein. Okay, actin is the most abundant protein that is present in eukaryotic cell. It is provide helps in providing shape, structure, and mobility, everything. But myosin is mainly present in the muscle fibers only. They helps in the contraction process. Actin is provided in both A and I band of the sarcomere, and myosin is found only in the A band of the sarcomere. Okay, and okay. Size wise, actin is shorter, actin is thin, myosin is thicker as well as longer. Okay, actin is globular, myosin is motor protein. Molecular weighting, since actin is thin filament, so we can assume that actin will be smaller in molecular weight compared to myosin. Myosin is more molecular weight. protein. Then actin filaments are more abundant than myosin. As we already discussed, they are most abundant protein in the eukaryotic. Myosin is less abundant compared to actin. There is one myosin for each actin filament. Um, Now, the surface of the actin is smooth, surface of the myosin is rough. Protein in filament, actin filaments are composed of three components, actin, tropomyosin and troponin. Troponin, again, there are three classification, troponin I, troponin C and troponin A. Myosin filaments are composed of myosin and meromyosin. This meromyosin is light meromyosin and heavy meromyosin chains are there. Actin cannot form cross bridges. Myosin is able to form cross bridges. Actin is not associated with ATP molecules. Myosin remains associated with ATP molecules. Okay, so please keep these things in the mind. Now, one end of actin filament is free while other is connected to the Z line. But in myosin, they are both ends are free only. So they are like staying in between these two. So the head domain of myosin, however, remain in association with ATP, but they are connected to the ATP because there is a ATP binding structure specifically at the myosin head. Sliding, actin filament is able to slide into the H zone during contraction. Myosin filaments do not slide into the H zone during contraction. Actin is present in muscle fibers. Microfilament is uh, cell membrane as well as cell wall, but myosin is only majorly found in the muscle cells. Actin interact with myosin. Myosin initiate muscle contraction by generating a force. How? By binding it to the ATP. Now let's solve more questions. In order to muscles to contract, they need calcium ions. Muscle cells have an organelle that stores the needed calcium ions. This organelle is called fascia, sarcolemma or sarcoplasmic reticulum, which is the organelle that stores calcium ions in muscle. In the video also it explained, I also said when we said that. So what is the correct answer? Can you please type the answer in the chat box? Then we can discuss. Which is the organelle that stores calcium ions? Thank you, Anbalagan Veeramuthu. I am sorry if I am pronouncing the name wrongly. Then you can unmute and say, okay, my name is this. Only one answer is there. Please, everyone, anything is fine. Thank you, Shreya. Yeah, can you please say how to pronounce it? Your name? Is it Anbalagan? Is it like that, or is there my pronunciation is correct or wrong? Okay, thank you. Thank you, Anbalagan and Shreya. So, what about others? Mm, Maivivi and Bandi Rachana. Can you guys also please type in the chat box what is the correct answer? Sorry. This answer already displayed. Yeah. So the correct answer is sarcoplasmic reticulum only. As we said, they can store calcium ion. Myosin cross bridges are extension of dash. Myosin cross bridges are extension of dash. Actin, myosin or troponin. Myosin cross bridges are extension of dash. Myosin. Shreya is saying myosin. Any other answers? Is everyone supporting? Okay, Anbalagana is saying actin. Okay. So let's see the answer. Should I go back to the figure or again my screen is stuck? Correct answer is myosin only. Myosin cross bridges are extension of myosin only because myosin can only form myosin cross bridges. Actin is not able to form the cross bridges. This will bind, but cross bridges are basically formed by myosin only. Okay. The membrane around a muscle cell is called. Option A sarcoplasm, option B sarcolemma, option C sarcomere. Membrane around a muscle cell is called. Thank you, Shreya. Any other answers or everyone supporting Shreya? Shreya is saying membrane around a muscle cell is called as sarcolemma. And Belagani is also saying the same. Okay, good. Yes, membrane around a muscle cell is called as sarcolemma. Good, correct answer. Actin filaments are attached to dash. Option A is Z disc, option B myosin, option C sarcolemma. Where is the actin filament attached? Is it Z disc, myosin or sarcolemma? Where is actin filament attached? No answer? Any answer is fine. Where is actin filaments attached? Remember the sarcomere figure that we just now saw? Yeah. So the correct answer is there, right? Yeah. Correct answer is Z disc only. Option A. Very good. Next question is troponin or tropomyosin complex is associated directly with which of the following element? Troponin or tropomyosin complex is associated directly with. So importance is to directly with which of the following filament. They are directly bind to which part? Any answers? And there is a helpful link in the clue also. Troponin or tropomyosin complex. Okay. Shreya thinks it's myosin. Any other answers or everyone supporting to Shreya? Anbalagan is saying thick filament. Okay. Let's see the correct answer. Correct answer is actin. Basically, actin has three parts, right? Troponin, tropomyosin, and actin. So they are basically always connected with actin only, right? And I said there is a clue in the option because thick filament and myosin is same only. Thick filament is the myosin filament, right? So they are basically same. So we can avoid B and C, and the correct answer is option A, actin. Okay. Moving to the next question. Yeah. Do you want to say anything, Shreya? You have a question regarding what? Ah, okay, okay. Yeah. What is the question? Hmm. Mesenchymal stem cells are differentiated into. Okay. So you have a doubt regarding that question. That's what you are saying. They have released the answers and explanation, no? Did you check that? Yes. That are the functional unit, no? Osteoblast helps in the formation of the so what happens? Individual units of cells are called osteocytes. Right. Osteoblasts help in the formation. So basically, it is formed from the precursor cells or stem cell. But stem cells are after differentiation. So that's why the question is they have produced and differentiated to what is the question, right? Osteoblasts still have the capacity of forming the cells only. They cannot differentiate. So after differentiation, the cells are called as osteocytes. Okay, they are the individual functional unit. Osteoblast always helps to form the new cells. So they are the precursor cells. They can divide and forms new cells. Then each individual cells after forming and differentiation that is called as osteocyte. Understood? Yeah, sure. Now next question is yeah, the binding sites for cross bridges are located on. Again, be careful, okay? That we discussed already the difference between actin and myosin. That time we discussed. The binding site for cross bridges are located on dash. Actin, myosin, troponin. Where is the binding site present for cross bridge formation? Actin, myosin, or troponin? What is the correct answer? Say is saying actin within the sarcomere. So, is my did my BD has any answer or you support Shreya's answer? Yes, correct answer is actin only. Binding site for cross bridges are located on actin. But actin has three different units, right? Actin, tropomyosin, and troponin. Then we have to look into is there troponin is there or not, right? Is the binding site is in present in which site? So the correct answer is I am really sorry. My screen is getting stuck like anything. Each part it is getting. Yeah. So correct answer is actin only because what are troponin? Troponin has sites for calcium, one for actin and one for tropomyosin. Binding site means the active site. Active site is present on actin only. So correct answer is option A. Correct. Next question. When muscle contracts upon stimulation, calcium ion binds to dash, which exposes the binding site for the myosin cross bridges to attach to. So first step as we discussed, it is the release of calcium and calcium binding to some point. So what is that calcium binding point? And after that, due to that binding only, the active site get exposed and as a result, myosin will able to form cross bridges. But question is, where is the calcium ion binding to? Actin, myosin or troponin? What is the correct answer? Is it acting? Yeah, good. Can you say which subtype of troponin it is? Which subtype of troponin? Any idea? If you don't know also, it's completely fine. We'll discuss, okay? Okay. So troponin C, troponin I, and so calcium is binding to, remember the C. So troponin C binds to calcium. When the crossbreeds bind to actin, they will pivot in such a manner to cause actin filaments to slide. This pivoting action causes the actin filament to slide. Option A farther apart, option B closer together, option C towards the Z line. What is the correct answer? Sliding filament helps or moves the actin filaments in which direction? Farther apart, closer apart, or towards the Z line? What is the correct answer? Any idea? Yeah, correct answer is closer together only. They helps the sliding to happen and pulls the actin to closer together or away, not towards the Z line, away from the Z line, not farther apart. It will always brings the actin closer together. Fourth question is calcium ions are released from sarcoplasmic reticulum to initiate muscle contraction and are reabsorbed upon muscle relaxation. Which of the following occurs if calcium ions are not completely reabsorbed? Next muscle contraction will be greater than the previous muscle contraction. Next muscle contraction will be weaker than the previous muscle contraction. There won't be a second muscle contraction until all the calcium ions are reabsorbed. What is the correct answer? If calcium ions have, if there is abundant amount of calcium ions, still calcium ion, then in that case, what will happen? A, B, or C? What is the correct answer? Yeah, correct answer is option A only. If calcium is there, it keeps on binding to the site and the cross bridges will keeps on forming, right? So the second contraction will be greater than the first contraction, right? So correct answer is option A. Good. Now let's move to the next question. Again, screen is stuck. Yeah. Sorry. Please don't look the answer. Hmm. In order to make ATP, adenosine triphosphate in muscle, creatinine phosphate transfers dash to the adenosine phosphate. Is it oxygen, phosphate, or adenosine? Simple answer. Even in the name itself, that component is there. So it's easy, right? What is the component? Oxygen, phosphate, or adenosine? Yeah, correct answer is B, phosphate. ATP can be made in muscles form from stored glucose. In muscle, glucose is stored in the form of dash. Glucose is stored in the form of energy, creatinine, or glycogen. What is the correct answer? Glucose is stored in the form of dash as in the muscle. What is the dash? Yeah, correct answer is glycogen. Lactic acid, a byproduct of cellular respiration, is typically produced during dash type of respiration. Which is the correct answer? Aerobic, anaerobic, or isometric? Lactic acid. This we have studied in plus one, plus two NCRT textbook. Yeah, it's produced in anaerobic respiration. Good. ATP is required for muscles to contract. Which of the following statement is false? ATP is used to get the cross bridges to bind to actin. ATP is used to get the cross bridges to disconnect from actin. ATP used to get the troponin tropomyosin complex to move in such a manner to expose the binding site. What is the correct answer? A, B, or C? So you can take the most suitable and correct option if you feel like everything is correct. Apt answer for the question. Correct answer is option C. Okay. So in the correct way, they will make the way of troponin tropomyosin complex to move and then expose the binding site. That's very after binding to ATP that is happening. So correct answer is option C. Which of the following correctly identifies muscles components in order from largest to smallest? So question is in the descending order. What is the largest unit and smaller units? There are three components only: muscle fiber, fasciculus, and myofibril. So, what is the smallest and what is the biggest unit in that? And choose the correct option. Any idea? Yeah, correct answer is option D. Good. Individual unit is myofibril. Myofibril join together to form muscle fiber. Then muscle fibers are joined together to form fasciculus. Space between two Z lines constitute the dash. Option A sarcomere, option B sarcolemma, option C sarcoplasm, option D sarcophagus. What is the correct answer? Space between two Z lines. It's not sarcolemma. Sarcolemma is the membrane, right? So the individual functional unit in sliding filament theory is the sarcomere. So two Z line in between the space is constituted as sarcomere where thin filament and thick filament are arranged. So the space between two Z line constitutes sarcomere. Option A is the correct answer. What is the role of tropomyosin in muscle contraction? Option A to release the troponin from tropomyosin, allowing the muscle myosin to bind to the actin filament, to release the calcium from sarcoplasmic reticulum, to aid in myosin caulking on actin filament, to prevent myosin from continuing to slide up actin filament. What is the role of tropomyosin? What is the role of tropomyosin? Yeah, good. Option D is the correct answer. It will prevent the myosin from continuing to slide up the actin filament. Basically, when ATP comes and bind it there, then they will stops and they will detach. So that's how they stop. Dash are the myocardial cells. Option A neuron, option B pacemaker cells, option C beta islets, option D nephron. What is the myocardial cell? Myo means muscles, cardials means heart. So there should be something related to muscles present in heart. That cells are present there. What is the cell present in hearts? Any idea? Hmm? Yeah, pacemaker cells. Option B is the correct answer. Myocardial contractile cells constitute the bulk of the cells in artery and ventricle. Pacemaker cells are highly specialized myocardial cells, whose intrinsic ability to rhythmically depolarize and initiate an action potential which is responsible for the basal heart rate. So, myocardial cells are pacemaker cells present in the heart. Arteries are made up of dash. Simple question. Arteries, veins, everything are made up of dash. Which type of muscle? Good. They are made up of smooth muscle. So there is especially in the blood vessel, it is known as VSMSC, vascular smooth muscle cells, which constitute the major layer of cells in the arteries and are critical to maintain the integrity of arterial wall. What is the prime function? Prime function of these cells in the adult individual is to contract and relax, thereby regulating blood flow to regulate target tissues. Which of the following is not a component of skeletal muscle force dynamics? Option A, calcium ion. Option B, actin myosin. Option C, troponin tropomyosin. Option D, knots of Ranvier. Which is not a component of skeletal muscle force dynamics? Not a component is the question. Good. Knots of Ranvier are parts of the neuron cells, not the skeletal muscle. So, correct answer is option D, knots of Ranvier, which are not part of the. Next question is, so correct answer is option D. Okay. Knots of Ranvier are not parts. In skeletal muscle, which of the following prevent interaction between actin and myosin? If that is not there, they will keep on interacting, but that structure makes sure that actin and myosin are not interacting. Option A, myotube. Option B, actin. Option C, tropomyosin. Option D, troponin. What is the correct answer? Yeah, good. So, tropomyosin prevent the interaction between actin and myosin. Okay. How they prevent? Because they are a protein that is wrap around the actin and prevent myosin from interacting with it. This prevent the muscle contraction until the proper signal arrives. When the nervous system tells the muscle cells to contract, calcium is released from sarcoplasmic reticulum. They will bind to the troponin C. As a result, the active site become visible and myosin can form the crossbridge. So please keep this process in the order in mind. Okay. I am not able to move the screen. That's why I am multiple time coming and doing this. Satellite cells are dash and that dash. What are satellite cells? Is they are stem cells differentiate to myocyte? Is they are fully differentiated cells that are derived from myocyte? Is they are fully differentiated cells which can differentiate to myocyte? Or they stem cells that are derived from the myocyte? What is the correct answer? Satellite cells are fully differentiated cells, isn't it? Satellite cells have a particular peculiarity. So, sir mentioned in the class, they can be used in the repair and injury. They are a very research interested cells types. So, will they be fully differentiated or stem cells? Correct answer is stem cells differentiate two myocytes. Okay. Actin, myosin are dash. Carbohydrate, protein, fats, and all of the above. Actin and myosin are dash. We discussed multiple times this. Actin is this, myosin is dash. What is the correct answer? Correct answer is they are protein. Actin is globular protein and myosin is motor protein. So both are proteins. Correct answer is option B. Okay. Length of sarcomere is. What is the length of sarcomere? Is it length of an I band or is it length of an A band or is it length of an I band or length of an A band or is it distance between two Z line? What is the correct answer? What is sarcomere? We discussed this in a different form just before now. Easy, easy. Yeah, distance between two Z lines. Good. Oh, my screen. Which of this is non-striated? This also we discussed in the beginning of the class. What cell type does not have any striations? Option A skeletal muscle, option B cardiac muscle, option C smooth muscle, option D rough muscle. Which doesn't have striations? Yeah, smooth muscle it doesn't have striation. Very good. Which of those has no actin? Option A A zone, option B I band, option C H zone, option D AI zone. Which does not have actin? So it must be some central zone. I showed one image also earlier, which doesn't have actin. Please keep the figures in the mind and remember connecting to that we can say what is the correct answer. Answer? Correct answer is H zone. No, no. Correct answer is H zone. H zone is the central zone, right? So, H zone only has thick filament or myosin. It doesn't have any thin filament. Within the A band is a paler region called H zone. Its name is derived from heller, which means brighter, named for their lighter appearance under a polarization microscope. H band is the zone of the thick filament that does not have actin. Correct answer is option C. Next question. In myofibrils, the band that contains myosin filament and overlapping end with actin filaments are called. Band that contain myosin filament and overlapping end with actin filaments are called. Both myosin and actin are there. So what is will be the band type? J band, A band, B band, I band. Which option is correct? Yeah, Correct answer is A band. Good. So what is A band? In skeletal muscle, A band or anisotropic band is the dark band observed under a microscope which contains the entire length of myosin as well as the overlapping region of actin. A band's dark appearance is due to the high density of myosin filament and overlap with the actin filament. So in some region we can see light and dark pattern as Sir explained. Why dark pattern? Because it's very thick filament. So less light will penetrate through that part. So A band is um, consist of entire myosin filament as well as some overlapping part of actin filament also. Next question is actin and myosin filaments are connected side by side using dash. So actin and myosin are connected to the Z line by this thing. So what is that thing? Option 1 titin, option 2 keratin, option 3 osteocytes, option 4 creatinine. What is the correct answer? Side by side connected by dash. Any answers? What is the correct answer? No. C is osteocytes, right? Osteocytes are cell types that are present in the bone, functional unit of bone. Correct answer is chitin. Okay, I will show you the figure. Then you will remember. Again, this is stuck. Oh, yeah. Correct answer is chitin. Okay, what is chitin? Chitin is span from misal disc to M line in sarcomere, essentially acting as the backbone of this fundamental unit of muscle contraction. Chitin acts as a molecular spring, primarily in the I band region, providing elasticity to muscle and influencing the passive tension. Sure. Sorry, in this figure it's not written, but you remember this actin and myosin are connected to the this viscer line by the structures called as titin. Okay. Now next question. The subunit of troponin that has higher affinity for actin. The subunit of troponin that has higher affinity for actin. Troponin I, troponin T, troponin C, or none of this. What is the correct answer? Correct answer is troponin I. Okay. Troponin I is the subunit that can bind to actin and helps hold actin tropomyosin complex in place preventing the interaction between actin and myosin in relaxed muscle. Okay, so remember troponin I bind to actin, troponin T bind to tropomyosin, troponin C binds to calcium. Okay, remember these three. In myofibrils, the bands that only contain actin filaments are called only contain actin filament. J band, A band, B band, I band. A band we saw in one of the previous question. It contains both myosin and overlapping part of actin. Yeah, good. It is isotropic band. Option 4 is the correct answer. Very good. So I band is the lighter appearing part as it consists of thin filament or actin filament. It's characterized the presence of only thin filament and is bisected by Z disc which angles this filament. Next question. Muscle fibers consist of myofibrils. In each myofibril, number of actin filament is almost dash the myosin filament. Option 1 equal to option 2 double, option 3 half, option 4 1 by 4. What is the correct answer? Number of actin filament is almost dash the myosin filament. Actin filament is more in number. So correct answer is double. Myosin actin filament is almost double the myosin filament. Okay. Specifically for every myosin filament, there are 6 actin filaments. Sarcomere length dash when actin myosin overlap. Option 1 increases, option 2 decreases, option 3 is unchanged, option 4 none of this. Sarcomere length dash when actin myosin overlap. What is the correct answer? Sixth question. Sarcomere length dash when actin myosin overlap. Fine, Freya. Sarcomere length को क्या होगा if actin myosin overlaps? It will decrease, right? So correct answer is option B. Because actin are pulled to the central part, so the length will decrease. Correct answer is option 2. Shortening of sarcomere or increasing the overlap between the filaments. So the sarcomere will decrease. Multi-unit smooth muscle is found in dash. There is single unit and multi-unit smooth muscle. Where we can see multi-unit smooth muscle is the question. Ciliary muscles, walls of uterus, gut lining, viscera of bile duct. Any guess? Correct answer is ciliary muscle of the eye. So usually where we can see multi-unit smooth muscle, they are like more control needed part. So in the ciliary muscle of the eye, light is coming, right? So pupil is dilating. So we need more control over there. That's why there is multi-unit smooth muscle is present. Okay. So they are present in the location requiring discrete control. And one is iris of the eye, walls of large blood vessel, airway of the lungs, etc. So this is like unlike single unit muscle, multi-unit is conduct independently and are not electrically coupled. allowing for more precise control over individual cells. Next question. If load applied on skeletal muscle equal maximum force that can be exerted on a muscle, velocity of contraction become dash even when the muscle fiber remains dash. What is the relationship between force and velocity? If you know that, you can answer that. High activated, low deactivated, infinite deactivated, zero activated. Hmm. But it's written maximum force, right? Your method of answering is correct. So maximum force means velocity is inversely proportional. So maximum force means velocity will be zero. We cannot say low because they have said maximum. And in the option 2, second part is wrong. It's written deactivate. Maximum force means if I am pulling something, if I am taking this water bottle and crushing it together, then it will get my maximum force is there, right? So it remains, muscle will remain activated, but velocity will be zero. So correct answer is option 4. Okay. So that is the F is inversely proportional to velocity, that is based on Hill's equation. Okay, keep that in mind. The velocity at which the muscle shorten decreases, and so muscle load increases, velocity will be decreases. This means lighter loads can be moved quickly and heavier loads can be require slower movement. Next question. Last question of the day. Okay. Myoglobin is mostly present in dash. Option 1 fast muscle, option 2 slow muscles, option 3 both fast muscles and slow muscle, option 4 none of the fast muscle and slow muscle. What is the correct answer? Correct answer is slow muscle. Myoglobin is more present in slow muscles. Why? Because they are present, that's why they are present as the like red color. So they are also known as the slow muscles are also known as float-rich fibers. So this due to this myoglobin, these slow muscles have a reddish appearance. And why what is the speciality? These have rich in mitochondria and are used for endurance activities. So please keep that in mind. Now, if you have any doubts, like earlier you asked, both of you, if you have any doubts, you can ask me, then I will try to solve it. Otherwise, we'll wind up today's session. So you don't have any doubts? Then can we call it a day? Yeah. Thank you so much. Please go through the assignment questions. Okay. Try to solve it every week. It will helpful for your exams also. Uh, can you please tell me, Shreya, where are you studying in? Where are you from? Where are you studying? Which course you are doing? Oh, you are from Odisha. Okay, which part of Odisha? I am studying in Aisar Barampur. So I am not from Odisha, but I am studying in Odisha. In which part of Odisha you are from? Oh, Balasore. Okay. Myvivi, where are you from? And which course you are doing? You can also type. Okay, microbiology, Namakkal. Okay, okay. Nice to meet you, both of you. My name is Harsha. Full name is Harsha Shankar SH. I am a PhD scholar in Department of Biological Science in ISER Perambur. Currently, I started my fourth year of PhD. Uh, I am working under the guidance of Dr. R. Salvi Bharata Vikru. My lab name is Ribonucleoprotein Biology. So that about myself. Okay, then see you in the next class. This week's my session will be on this Friday, 6 to 8. Last week I could not able to take on Friday because some emergency happened. I was coming back to Odisha from Kerala and my flight got postponed. So I could not able to take due to the travel change. That's why I postponed it on Tuesday. This week it will be on Friday. Okay. See you then. Thank you for joining. Bye bye. Hope it helped you. Bye bye. Take care. Yeah. Thank you.